This is our latest high-density microelectrode array chip. It's called MEA1k. You see the chip bonded to the green board. A glass ring glued to the board to form the culture chamber. The epoxy covers the pads and bond wires while exposing the electrode area to the cells and the culture media. On this chip, we can culture rat cortical neurons. We can also put the brain slices on the chip and perform recording and stimulation. The chip is then connected to the support board. This board has voltage regulators and digital to analog converters for generating voltage references. The board has a small FPGA on it which receives the commands from main FPGA and sends this command information to the digital to analog chips on the board. The received parallel data from the MEA1k chip is sent to the main FPGA board using the LVDS connection. On this board, we also have the connections for probing and testing the chip. For the main FPGA, we are using Vertex 5 from Xilinx. We can use this FPGA to perform signal processing, such as spike sorting or closed-loop stimulation. The data is then sent to PC using Ethernet connection. On the PC, we have the software to save and monitor the data. And we also use this software to send the control and configuration commands to the chip. Now, let's look at this software in more detail. So, this is the software used for interfacing to the chip. On the left side, we can see the real-time data for 1024 channels. The red indicators show when the spikes have occurred, which is the result of a spike detection algorithm. On the top right of this window, we can see the adjustments for the number of the channels to be monitored. We can change the number of boxes to be displayed, as well as the number of channels shown in each box. On the middle, we have adjustments for time scale. The sampling frequency of each channel is 20 kHz, which means every 50 microseconds we receive a new data for each channel. By changing the time scale, we can change the number of data points that we want to monitor. On the bottom right, we have the range adjustments, which can be adjusted based on the received signal amplitude. Now let's look at the stimulation interface. On the stimulation panel, we can control the amplitude of the stimulation signal. This is done by sending the configuration command to the digital to analog blocks on the MEA1k chip. For stimulation, we use positive then negative biphasic pulses. We have the possibility of voltage and current stimulation, as well as auto-zeroing feature. Now let's look at the full electrode area. On this window, we can see the whole chip area and the position of the electrodes chosen for recording. Now we can load the image taken from microscope. Here we can see the microscope image of the cultured neurons on the background of the full electrode area. The small purple dots represent the location of electrodes, and the labeled cells are shown in green color. We have a total number of 26,400 electrodes on this area, and we have 1024 simultaneous recording channels on the chip. Therefore, we can configure the array and choose which electrodes to be connected to the recording channels. Let's say we are interested in the activities in these two areas. So we configure our high density recording to be focused around these two areas, as configured in this case. Now let's remove the background image for better visualization. Now we can see the neural activities on these areas. We also have the feature of monitoring the footprint. This feature gives us some information regarding the places where the neurons are located. Due to the high density of the electrodes, each neuron's activity is detected by several electrodes, which provides the possibility for more accurate spike sorting. There are also other features for visualizing the spiking activities, such as this current spiking rate, which gives us information about the number of spikes per second. We can also see the raster plot, which shows the overall recorded activities, such as spiked bursts, over a longer period of time, in this case 1 minute.